IIT Madras. He's he's a person who admires us all because he usually delivers lectures in a fruitful way, and his lectures are always welcomed by the participants of Institute of Innovations. And I request all the participants to keep in mind some points regarding the what they should do during the lectures. I request all the participants to mute your video and audio during the whole session. And if you switch on the video and audio, you will be maybe you might be disturbing some other persons because it will create some disturbances. So if you have any doubts, you can feel free to raise your hands, and your questions will be answered at the end of the session. And at the end of the session, you can switch on the audio and you can ask any doubts if you have to Professor Praveen. He'll be readily available to you to answer all the questions. With the permission of Professor, I am saying this. And I request all the participants to go and check your mail, because we had sent the Google Calendar notification to you. So please accept the invitation, Google Calendar invitation. So once you accept the invitation, you might have you can directly join to the meeting, because every day we are admitting you. You are requesting to let into the meeting, so we are admitting you. Instead of doing this kind of thing, you can directly join the meeting if you accept our invitation. Like I'll show how to do that. There's some network lag. Just wait, I'll show how to do that. Professor, sorry, I'll take some other couple of minutes, Professor, please.
No issues, Professor. You can kindly.
Yeah, thank you. Mr. Dhanush, can you mute? Yeah, thank you.
Yeah, hello, sir.
Yeah, please mute your mic.
Okay.
Yeah, thank you. So some of the people raise their hands. Shweta Selvaraj, do you have any doubt to clarify? Miss Sandhya, they had raised their hands. If you have any doubts, feel free to ask us. So there is some issue in my system. I wanted to show you how to join seamlessly. So yeah, no, it's okay. My screen is visible. Yeah. So you might have received a mail like this. This is the invitation. AIML course daily from 7 p.m. to 8 p.m. You might have received a Google Calendar notification like this. If you didn't receive the email, if you didn't saw the email in your inbox, just check your email using our email ID. Our institute email ID is praveen@innovationinstitute.co.in. So check your email box using this email ID. That's praveen@innovationinstitute.co.in. So this is the Google Calendar invitation. So a mail like this will load in your window. So once the mail is open, you just click this yes. That's all. If you click this yes, and then you ask to join the meeting, you can directly join the meeting. There is no issues. There is nothing like we have to admit you like that. So you just check your mail using our email ID, praveen@innovationinstitute.co.in. So a mail like this will open in your window. So in this, you just select yes. If you select yes, you can directly join the meeting daily without any disturbances. So you could not. There is no need for waiting for the admin to admit you in the meeting. So I request all the participants to do this after today's session. So kindly note. Kindly note that this is the same link is for the all days. So there is no new link for any day, and so this is all from me. And if there is any instructions from us, I'll be letting you know time to time. And I request all the participants to save our institute's phone number. I'm typing my phone number, institute's phone number, in this chat box because. 488-773-165. So we don't use to create any WhatsApp groups, but we will be sending the messages by broadcast messages. So if you save this number in your mobile, you can get the message. If you didn't save this number, this message will not be delivered to you. So I request all the participants to save this number, which I had mentioned in the chat box in your mobile phone, so that you will be receive all the communications time to time, so that you cannot miss any communication from us. So this is all from me right now. So without without wasting much of time, I would like to hand over the session to Dr. Praveen Kumar sir. Sir, please, sir.
Sir, thank you very much,
Yes,
Professor Praveen sir.
Yes. Yes, sir. Sir, there is a question in the chat box, sir. We didn't. Okay. Anjana, I had added you your name in the broadcast group today evening only because your name was saved in some other events registration number. So that registration number didn't showed your name in that broadcast group when I added. So today I searched that and added your name there. So you will be receiving all the recordings hereafter. And Sastika Ghosh, we had sent the recorded sessions yesterday and day before yesterday. Might be you didn't saved our number, so that you did, so you had not received our messages. So I request you to save the number. However, I just a minute. So this is an example. I'm just showing the message which we had sent from our institute by broadcast message. So Swastika, as I said, I didn't save our number, so the messages were not shown to you. I had shared my screen, just see, it had sent the messages to you. It was not delivered to you due to you have not saved our number. So please save our number in your device so that you will be receiving all the messages. So however, I'll be sending the previous sessions recordings once again to all the people. So that's all. Thank you, Professor.
Thank you, Professor, for providing a brief introduction and giving some kind of suggestions to the participants to how to start your sessions and how to receive the videos. Thank you very much. Am I audible, Professor? Professor, am I?
Yes, sir.
Yes, Professor.
Yeah, good. Good evening to the participants and the coordinator of this internship and certificate course on artificial intelligence and machine learning approaches in drug design, drug and vaccine design in cancer. So today, my topic is about the methods of drug design. And as I earlier mentioned, so I thank once again Professor Praveen Kumar Ramanujam sir, who has given me an opportunity to provide this kind of certificate courses. I already have trained nearly nearly some 6 to 7 certificate courses. The collaboration of Institute of Innovations, Tiruvannamalai. So now I just start with the session. Before going to the methods of drug design, okay. Yesterday I was explaining you something about. Can we recap what exactly we discussed in yesterday? Later we can come to your new topics. So just I want to have an interaction with you participants now. What are the topics we discussed in the last session? Topics discussed in last session. So type it fast. So very good. Drug development and discovery is on the first session, isn't it? How about on yesterday's session? I got instant response from Veera Rahul. Good. But what exactly? Very good. Target identification. I got the answer from Sahana, and modeling and validation also got from Varshini. Good. So we also discussed something about target deconvolution. So what is the difference between target discovery and target deconvolution? Very good. Also, I got an answer from Tanvi that we also discussed about drug elucidation. Okay, so whatever you find something new. Okay, so I think everyone, all the students are displaying the kind of technology terms, the terminologies used in drug design, and what else? Very good. We also discussed about database for targeting the drugs instead of simply restricting up to targeting the receptors. I mean, how to download the target receptor, 3D structures, and how to validate the structures. We also discussed something about identifying the drug targets. Okay, we also seen a therapeutic target database, etc. Very good. So we have also seen the difference between traditional based drug design and structure based drug design. Very good from Varshini. So molecular modeling, we have seen methods of molecular modeling of proteins. We have seen homology modeling. We have seen threading as a second method, and third is the ab initio modeling. Just we have seen only the methods, and I also shown you corresponding tools from XPASI web server. You remember? So if you receive the recorded video, those who don't have a repository, you can watch all this. That is the methods of molecular design, molecular modeling of proteins. We also opened, we have also seen some softwares, isn't it, for visualizing a 3D structure. Can you tell me what is a visualizer? I have shown you to see the 3D structure of proteins. What is the software I've used for visual? Very good, BioVia. But BioVia is a package. If I ask you what is the software used for plotting the Excel, plotting the graph sheets in MS Office, you tell MS Excel, isn't it? So BioVia is a package, but you have so many modules inside. Like if I ask you what is a software used to draw the graphs, okay, in MS Office, you tell like MS Excel. Likewise in BioVia, Discovery Studio, good, of course, but in BioVia, Discovery Studio is a package still. BioVia is the name of a company. Discovery Studio is the software name. But what is a module? Even Discovery Studio is a package. BioVia is a company name. Can I type the answer? BioVia DS Visualizer. Okay, the name of the software is Visualizer from DS. So a product from BioVia. Okay, we also seen very good the databases like PDB-Sum. Also, we have seen Swiss model, an online tool. Okay, so a tool which is used for predicting the 3D structure of proteins, where if the structure is not known, but if the sequence is known. Okay, so these are we have seen. How about validation? How we can validate the quality of a protein structure, especially stereochemical quality of a protein structure predicted from any of the protein prediction methods. How to validate the structure of a protein? I told you you can use. Very good. The answer is Ramachandran plot. I got answer from a few participants. Good. So Ramachandran plot is to validate the stereochemical quality of a protein. Good. Uh, very good. So many are answering. I think this is what we have discussed in the last session. Okay. But till now, whatever we have discussed, we have seen how to model the proteins. But how to model, because we have discussed about target modeling, analysis and validation, we have seen only up to the receptors. But we can also able to model ligands. Okay, so that will be seeing subsequent sessions. Okay, let me start the session 3. The session 3 is about methods of drug design. There are plenty of methods of drug design. Okay, and nowadays we are focusing on artificial intelligence and machine learning approaches to discover new tools in each and every sectors. Okay, in designing drugs or vaccines for types of diseases, including rare diseases. Okay, let me just start with a slide like, related to the today's session. How researchers discover the drug for a disease? Okay, so how the scientists can go for discovering a drug for a disease. Yesterday, we also discussed with an example, like if you want to cure fever, if you want to get rid of pyrogens from in your blood, okay, we generally go for choosing acetaminophen, isn't it? Many people won't go for doctors if they have a normal fever. Simply, they can go to the dispensaries and they can ask for the acetaminophen. And even the persons who sell the medicines immediately can give you the tablets, isn't it? Without any kind of restriction in showing the prescription from the doctors. But the same, is it possible if a patients are suffering from cancer? If a patients are suffering from cancer, whether we can able to get drugs from the normal dispensaries, common dispensaries nearby your home? No, we can't, isn't it? Only the cancer specialist hospitals and the dispensers very close to that, with a proper license, only they'll be getting these kind of medicines. That too, the medicines can be supplied if only if the doctor is prescribed, isn't it? So how these drugs are discovered actually? If it's not a common drugs for treating a common cold or fever, so how these drugs are discovered? For this, I already told you, especially for cancer, we need precision medicine design. Just like that, you can't able to discover it. Disease. Discover a drug for a disease like a common fever. Okay. So much. So we should understand the genes. We should understand the protein targets responsible for the specific type of cancer. We have complications in cancer, isn't it? So if a person is diagnosed with cancer, might have the cancer has spread to various tissues, gone through the metastasis, and might whatever organ they feel pain, they have to check from the pain, and they might have diagnosed with a different type of a cancer, isn't it? So if a patient is suffering from a kind of a pancreatic cancer, definitely he won't feel any pain in the pancreas. But the pain might have circulated, might have generated from the nearby organs. Okay. So just like that. Okay. There are so many kind of cancers where you can find something mysterious. We can't able to find. The prognosis and diagnosis is most important. Okay, so if you can able to find it using the proper markers, then we don't have any problems. But still, with the prognosis mark, whatever you identified as a gene or a major hub protein which is responsible for cancer, you have to target accordingly. Okay, so you not only have to target a single protein, but the hub protein interacting with the interacting proteins. That's what we have discussed in the last two sessions, isn't it? So If you see this kind of a drug design in cancer, it's not like a simple, it's something complex. Just even if you see from the traditional to the structure-based drug design, okay, still we find more complications in structure-based drug design. Okay, even if you have structures, we should also think about network pharmacology and other approaches, especially for cancer like diseases. Okay, well, I think even this we had discussed in the previous session itself. So we have to revolutionize the drug discovery in silico, not just by understanding the structure of a ligand and receptor. That knowledge is not just merely sufficient for drug discovery for drug design in cancer. Okay, so this is what I want to mention. Then, what are the basic methods we can use? Okay, then we can come step by step. Okay, so what are the basic methods? Generally, the methods we use are the virtual screening methods, of course, to discover the drug for a disease. We can't able to simply restrict with a single drug, and a doctor can't prescribe a single drug for treating a cancer. He has to go for so many drugs in a combination. Even if it is a chemotherapeutic treatment, the patients feel more pain, tremendous pain. Okay, it's not like taking a tablet for a common fever. The patient suffering from cancer, even if they are prescribed to take oral medicines, these medicines give like a significant pain to the patients. Then we should be keen. We should show more interest to develop precise drugs for the cancer. We should not randomly discover drugs. So, who are doing research in the cancer, you should alert on, you should keep alert on all. Like you should not give any choices. Okay, you should give only the correct kind of information if you go for drug design or development for this type of disease cancer. Okay, let me go to this virtual screening method. I told you we are not just going to check the binding affinity of a single drug, but we are going to check with the multiple drugs. Then only we can understand whether these drugs are targeting the target protein receptor or not. Okay, if you go this if you go to this kind of a method called virtual screening, if you start with the 3D structure of a target, that is a target protein, we have two types of methods. Okay, we can use either ligand-based methods or structure-based methods. In ligand-based methods, if the ligand structure is known, okay, through the clarity, like if you find one drug is efficient in treating the cancer, and if you want to find a most efficacious drug with the backbone of the drug, if you can able to alter any functional groups and in the existing drug, and if you can able to find the drug whatever you produce is more efficacious than the pre-existing drug, then these drugs can be overdated with the pre-existing drugs, isn't it? So each and every day, many scientists are working on to come up with a new kind of a drugs. Okay, if they have a kind of a similarity kind of a searching in the databases. So in the database, so you also have a tools. The tools can able to search, okay, through the artificial intelligence approaches, and they can give you similar kind of a ligands, a 3D structure of a ligands. You can also check binding of these ligands, okay, these ligands with the target receptor, and you can check how much binding affinity is shown with the, and you can compare with the pre-existing drugs. There is also one more kind of a method called pharmacophore mapping. Okay, the pharmacophore mapping is also a kind of a method. If you know the 3D structure of a ligand, okay, you can able to check by incorporating the functional groups in the major backbone of the drug, and you can check how efficient these functional groups are attaching with the protein receptor, the disease protein receptor. And if you have both actives and inactives, actives, we mean like an active pharmaceutical ingredient. Do you know what I mean by active pharmaceutical ingredient, API? Like if you take a paracetamol, yesterday I was explaining you, this is also called as acetaminophen. Okay, commonly called as acetaminophen, which is used for treating the fever. If you go to the Japan, if you go to the US, if you go to UK, okay, wherever you go, acetaminophen is a common name. But paracetamol, here we call it as a paracetamol. In US they call it as a Paracet, and somewhere in the Japan they may call it as a Paramol. So we have different types of names. Okay, that's what I'm telling. Acetaminophen is a common name. Okay, now I was explaining you something about API, active pharmaceutical ingredient. If you take a paracetamol of 500 mg, okay, if you have just 10% of the drug. I told you on yesterday itself, if you want to take a drug for a cancer, for a fever, you have to take 3 doses of acetaminophen, isn't it? As the residence time distribution of a drug in our blood is just 8 hours, not more than 8 hours. So the doctors, any doctors, prescribe this acetaminophen for 3 doses. Okay, unless they get relieved at least for a day. At least for a day, if they get relieved, or even if the temperature is decreased for more than 12 hours, then they can tell to stop taking the drug, isn't it? Okay, so if you see this kind of acetaminophen, even in 500 mg, okay, you have just 10% of a real drug is 10%, but the remaining 90% are surfactants, fillers, viscosity enhancers, and some kind of sugar binders. So many kind of ingredients are there. We have just 10%. I mean, only 50 milligrams. Only 50 milligrams from the 500 milligram of paracetamol is going to target 6 liters of blood. I told you, average 75 kg male or female can able to have 6 liters of blood in circulation in our body, isn't it? But the 50 mg API of your paracetamol, the acetaminophen tablet, is going to target that. What is it? Receptor I mentioned you on yesterday. Can we have a kind of interaction? Yeah, very good, Sangavi. So we have more 90% additives, excipients. Yes, good. The answer is COX-2. I think she is more active. Okay, COX-2 is the enzyme, okay, which is the major target receptor. We may target few more receptors, but this is the major target receptor I mentioned you on yesterday. Okay, and now if you see, if the actives are known, then we can go for this kind of similarity searching or pharmacophore. If the actives are unknown, if you have more inactives, better move to the machine learning methods. You can use the machine learning methods and discover a new tool if the tool is not available for the specific type of disease, whatever you're going to come up with a drug. Got it? If you have some inactives, I mean inactives or the kind of a drugs which we generate from the computational part. Whatever structures we generate from the computational part, these structures are also deposited in the database. But in real time, these drugs are not came to the market. But even if you want to check with these kind of inactives, we can make it out. We can make it out by using machine learning methods. Got it? So, and if you have a kind of a known drugs, okay, if you like, for example, anti-malarial drug, initially they tried with anti-SARS-CoV-2, isn't it? They check whether it works like an anti-SARS-CoV-2 drug. Okay, like repurposing. Okay, the repurposing of drugs, we call that kind of using drugs for one virus targeting for the another virus. These are repurposing methods. This can be done if you know the structure based. So if you have a structure for the SARS-CoV-2, if you try to check the binding affinity of the already existing some related kind of a drugs with a new kind of a target receptor, then we can come up with whether this is if it is having a good binding affinity, then we may conclude this may be a efficacious one. Then we can try with the in vitro and in vivo studies later. Okay, followed by clinical studies. Okay, so mostly how I started, the major method is the visual screening method. So if you go for the ligand based, okay, we can use pharmacophore modeling and QSR. And if you're structure based, we can go for molecular docking and MD simulation approaches. If it is hybrid based, like either if you have a ligand or a kind of a structure, then better go structure. I mean the structures of both the ligand and receptor. Structure based is both if you have a structure of both a drug as well as the target protein receptor, then it can go for this docking and MD simulation approaches. If you have either the ligand alone or if you have the ligand structure and receptor based structure, hybrid based, you can go for pharmacophore based docking approaches. Okay, let me cover in detail about at least a few important methods in drug design. Vaccine design, I can see in the last 4 class sessions. Now we can go for the drug design. In drug design, we have a drug likeness prediction. Okay, so if you want to check a plant compound, if you take a plant, yesterday I was explaining with an example, even if a particular plant is beneficial to treat a specific disease. For example, if you take cold, cough, okay, so the best, if you ask me which is the best kind of a plant or a tree, I can tell the neem leaves. The neem leaves are very good in treating not only the bacterial origin kind of a cold and cough and other kind of symptoms related to that. It can also be able to target viral kind of diseases, isn't it? Neem leaves have high potential. So in earlier times, they go for these kind of neem churnas. Okay, now if you see this, okay, not all the compounds extracted from the neem can able to have the beneficial effect for treating the disease. Only few compounds are more beneficial. So you just extract. That's what we came with a structure-based drug design, isn't it? So whatever compounds you get from neem leaves, for example, if you have 1,000 compounds, okay, there are nearly 1,000 compounds if you have in neem leaves. Okay, but if you want to check which is the best compound, which is targeting a particular protein responsible for generating the cough or cold, okay, then you have to predict initially the drug likeness for the compound. The compound may also have a toxic effect. Few compounds, whatever we extract from that, we should not take that because it's we have a minimal percentage of composition. We are not bothered about that in neem leaves, but even we should also, if you want to target specifically, especially for the diseases, for the cancer-like diseases, we should just isolate a compound, not the entire extract. So better go for drug likeness prediction using these kind of a tool, small inspiration. Even drug likeness nowadays, we can also able to find the data even from the NCBI PubChem. In NCBI, is the largest web resource where you find plenty of databases, okay, to download protein sequences, nucleotide sequences, structures, and also we can download the 3D structure of chemical compounds, drugs, inhibitors, all these compounds, 3D structures, 2D structures, and information more on that, and even drug likeness information, all these things we can able to find even in this database. Okay, NCB PubChem. Okay, even the structures of the chemical compounds and drugs are validated using the wet lab techniques. Even that information is also available in the PubChem. If you go to the ADMET prediction, that is absorption, distribution, metabolism, excretion, toxicity. This is what we call it as a pharmacokinetic property parameters. These pharmacokinetic parameters prediction can be done using Swiss ADME. This is a freeware. Pre-ADMET is also a freeware. So most of the tools, whatever listed, are the freewares. Okay, we can just you should have an internet to just log in to get inside the homepage of these websites, and then simply give the SMILES format, that is 1D information of a chemical compound. If you give a 1D information, obviously you can get more details about the drug likeness and other kind of characteristics that were what we call it as a pharmacokinetic properties of a drug. And if you see the third method, that is quantitative structure activity relationship. And the P stands for the property relationship. There are so many kind of properties. Even you can under properties you can find in the subsequent slides you find more detail about the properties. If you see this the freewares, we can use Galaxy, but DSQSL is a commercial one. Okay, it's a commercial one, but it works very efficiently. Pharmacophore modeling, pharmagist, pharmamapper, molecular modeling. Yesterday I was explaining about you the Swiss molar nitizer. Anyone can able to try these softwares. Okay, Swiss molar nitizer and molecular docking. The discovery should elegant fit auto dock. So maybe for these softwares you need a kind of a tutor. Even in our upcoming sessions, I teach you how to go for docking using Autodock. Okay, but these softwares are easy. Anyone can go to this website and simply paste a protein sequence and get information on that. Even these softwares, MolSoft, simply you should know what is the input sequence you have to feed. Okay, if you feed the sequence, obviously get an information. And if you see the method inverse docking, also called as reverse docking. If you in Google, if you type like acid space for inverse docking or reverse docking, it the home page shows in the first link. Just click the inside and simply paste a kind of an input, you get an output. All these things simply you have to open a website, paste a single input and get an output, but you should know how to interpret the results. Molecular dynamics can be done using the Bromax and AMD. For this, you have a high computational power. Okay, if you have a computational power, you can work on MD simulation. The participant, the newly joined participant, kindly mute. Yeah, I think some interesting movie is going on with a participant. But see, kindly take care. Even the other participants are on. Can I remove her? Okay. So participants kindly mute. Okay. We have very less time to complete. Still, till now I didn't go on to the to show the hands-on experience. Okay. Yeah. Now I can go across the read across approach can be done using the ToxIt, all tox rate, and the gene expression profiling and biomarkers development. We can read through the cytoscape visualizer. So most of the softwares, whatever is shown, 90 to 95% of the softwares are shown are freewares. Anyone can able to train your laptops itself. So only the Chromax, you need a high computational power. Don't try with the laptops. Only you should use a kind of workstations or servers to work on it. Okay, visual screening can be done with the Pyrex freeware, and machine learning technique can be done with DeepMiner and Python programs. If you see the drug likeness prediction, we have totally 4 properties to check whether your compound is working like a drug or not. Okay, the first property is the molecular weight of the particular compound. That should be less than 500 gram per mole. And if you see the second property is partition coefficient, log P, which shows you something about blood-brain barrier information. Whether a drug is a lipophilic or a hydrophilic, this information can be determined using the log partition coefficient. If the log partition coefficient should be less than 5 generally. And the third property is the hydrogen bond acceptor, which should be less than 10, and hydrogen bond donor should be less than 5. Okay, so these are the 4 properties of Lipinski rule 5. Why we call it as a Lipinski rule of 5? Because all these properties have in multiples of 5. The values are in multiples of 5. So we call this as Lipinski rule of 5. But according to Lipinski rule of 5, which is a drug likeness filter, if a compound behaves like a drug, it has to satisfy any of 3 of 4 properties. From 4 properties, if any 3 properties are satisfied, still we can tell the compound behaves like a drug. Okay, but what are these 2? These 2 are an additional. Rotational bonds less than 10, and polar surface area should be less than 140 angstrom. 140 angstrom. Okay, so if you see this kind of these 2 additional parameters, these are linked with a few more additional filters like Lipinski. Like Lipinski, we have Weber and few more kind of other drug likeness filters. Okay, in this in that kind of a filters, they have utilized even these properties. But see the wonder, even these properties you have multiples of 5. Okay, but don't match with the Lipinski rule of 5. The Lipinski rule of 5 is just about mentioning about the 4 important properties. And if you see the software, MolSoft, MolInspiration, and Swissaria, you can use these freewares to check all these properties. And drug likeness, in addition to the checking the 4 properties, what I mentioned in the Lipinski rule, we can also use a kind of a graph from MolSoft. If you see this graph for the drugs, okay, we can find nearly minus 1.2 plus 1.5. And in the green, which are non-drugs, this is having some other range. Like a binomial distribution, you can find a curve. If your drug, here my drug is lying in this red color line, which is in the blue color region. So I can tell still my compound is behaving like a drug. Okay, so through the graphical representation, we can mention that even though they are using some kind of a correlation to determine a drug likeness score, we got like minus 8.2, which is lying in this blue color region. So I can tell still our drug is, our compound is acting like a drug. And we have a property called ADMET, that is in addition to the absorption, distribution, metabolism, excretion of pharmacokinetic property, there is also one more property called liberation. The toxicity they didn't show, but toxicity is obviously is needed. Okay, if a paracetamol, we know that we have to take only one paracetamol but for a day we have to take 3 times, isn't it? So on forenoon, afternoon, and night. These are the generally recommended sessions to take this kind of a drug, isn't it? But if these 3 doses, if a person is taking in a single, instantly in a single moment, what is going to happen with the patient? You know, the patient will face side effects. The side effects like blurring vision, some kind of inconsistent or some kind of feel, which even if a patient is taking more than 3 tablets, okay, like if they are taking 5 or 6 tablets, they may lose their life. So that whatever drugs we take, okay, even it can be a paracetamol, we should take only up to a certain dosage, or however they prescribe. The concentration and dosage patterns or strictly has to be followed. Okay, so if you see this kind of a oral route of administration, that is taking a drug in oral route, initially the liberation is happening in the buccal cavity, and absorption. The absorption is happening in the blood, and distribution is happening in the tissues. Like for example, a patient is suffering from a pancreatic cancer, and if they are taking a kind of oral drug, and if they want to treat at least a kind of a pancreatic cancer, then whatever drug they take, okay, from the blood, it has to distribute to the pancreas. Okay, and if they want to treat a kidney, okay, to remove the kidney stones, obviously we have to distribute to the kidney. So the major drug has to reach the kidney. It should not reach in any other organs. But you know, we have an organ called liver, the largest organ, of course, next to the skin, where in this organ you find plenty of enzymes, cytochrome P450 enzymes. These enzymes play an important role in metabolism and other kind of activities. Okay, so the major role of liver is metabolism. I think many know that. So even whatever drug you take to treat to target any kind of an organ, nearly 50% of the metabolism is happening in the liver. So only the 50% is going to liberate, okay, distribute in the desired tissues, of course. Okay, so even the metabolism pattern is most important in the liver. Okay, even if it is another distribution, after the distribution we go to the metabolism, but even it reverses. It also comes in a cycle. So even we should know how much metabolism is happening in the liver, and the excess drug is excreted not only in the form of urine or feces, but the excess drug can be can be excreted in the form of sweat and other body fluids. Got it? So this is something about ADMET, the pharmacokinetic properties, okay, which we can able to determine even through the computational online software tools like SwissADME, QuickProp, StarDrop, MOE, KNIME, Notes, these stocks. So not only we can able to determine all these ADMET properties, but we can also check the toxicity of the drug. So we're fixing 500 mg of paracetamol, and that to 10% API is there. How we fix it? We can through determining the toxicity. Even the toxicity can be determined in the computational tool itself. Okay, no, not necessary to check with the in vivo or in vitro, the clinical studies. We have at least a kind of a tentative kind of a dosage determined from the computational studies. Later we can check it. Okay, now if you see this kind of output generated from the Swiss ADME, which is the tool which gives you not only about the ADMET characteristic, but also something about drug likeness and few more properties. Okay, this is a freeware. Simply go to the swissADME.ch. Just you have to provide the SMILES format. This is the 1D information of a compound or a drug, whatever you're going to feed. Just feed a 1D information. This we can fetch from even NCBI PubChem database. In NCBI, we have a largest database for a 3D or 2D structure of chemical compounds, drugs, inhibitors, etc. So you can get this kind of a single line information of that SMILES format and paste it. You get detailed description of that kind of a compound. Okay. So there is much to explain about this. Okay. But let me at least take you to some of the websites after completing this. Now, pre-admit, like however I'm going to the, I've gone to the Swiss ADMET. You can also use the pre-admit, which is for predicting drug likeness. Also to check the ADME prediction, that is whatever I mentioned, pharmacokinetic parameters. In pharmacokinetic parameters, we can go for calcium, the CaCO2, MDCK, BBB, blood-brain barrier, the plasma protein binding. So these are some of the things which is most important. Okay, to determine the pharmacokinetic properties and the toxicity prediction. Not only we can check with the AIMS test, but also in the rodent carcinogenicity assay. QSAR. These are the next method. Quantitative structure activity relationships. So if the activity of few compounds are known, and if you want to determine activity of an unknown compound, okay, initially you have to develop a training data, you have to develop a training model with the known activity of the compounds. Then from the training data set, if you have a test data set where you don't know activity of a particular compound, you can able to get an information from this QSAR method. You know, it's a purely a machine learning technique. It just looks like a neural network. Okay, so you have to develop a training data model, and then from the test data set, the particular property can be determined. Here in this case is activity. The property is activity. So property is a general term, and in the property, if you are determining an activity, we call it as a QSAR. So instead of activity, if you are relying on to predict reactivity, QSRR. And if you're predicting chromatography relationship, you know, there are some group of compounds in a single plant. You know, I did my research with in ashwagandha. In ashwagandha, we have steroidal lactone compound called vithanolides. We have 35 different vithanolides in a single plant. So all these vithanolides have a slight variation in the structure, slight differentiation in the structure. So even if you want to distinguish the chromatograph, it's very, very difficult. Okay, but they have some kind of a distinguished information, isn't it? So this kind of a chromatograph relationship can be studied using the QSCRs, and toxicity relationship in QSTRs, and electrochemistry relationship QSCRs, and biodegradability relationships QSPRs. There is a next method called pharmacophore mapping. Here, if you know a lead compound structure through the functional groups, okay, we can modify the functional groups. We can attach and detach the functional groups, and we can check which is the best lead compound with a particular functional groups. So we should know which is the best to target a particular receptor. Okay, if you want to target a particular receptor, first you should know a known drug, and from the known drug you can incorporate some functional groups, and then you can check the binding affinity, and if the binding affinity is more, then obviously you can tell that's the base compound where derivative we derive from the known drug. This can be done with the pharmacophore mapping. So for in this, you should what kind of parameters are taken in consideration? Steric and electronic features are taken in consideration to check in the pharmacophore mapping information. So actually, Zinc Farmer is one of the software, and these are few more tools used for predicting a pharmacophore. And if you see this kind of an output from Galaxy QSR, the previous one, okay. So we are from the training data set, okay. I have developed a kind of a linear regression model, okay. So predicted versus activity, I predicted a kind of a training data set model. And now if I have a kind of a drug, okay, this drug, okay, just if I predict if I have a drug, the activity is predicted from interpolation. Here I got an activity of a drug nearly as 8.3. So you have to simply get a kind of, you got it, how I am telling. So just if you take it to the straight, okay. And now if you want to predict the activity is around 8.3. Okay. Now go to the molecular modeling. In molecular modeling, yesterday we have discussed 3 methods are there. Okay. So for this, you have tools. I don't want to waste time on this because yesterday we have gone through that. And I also told you the stereochemical quality of a protein can be estimated from the Ramachandran plot through identifying the number of favorable residues. The favorable residues should lie only in these regions, in the dark green regions. If the amino acids are lying somewhere here, these are unfavorable residues. Even you have, sometimes you have residues somewhere even like this. So 1, 2, 3, 4, 5 are unfavorable residues. Okay, so this is how we can determine what are the favorable residues and unfavorable residues, and how you can predict the quality of a protein using the Ramachandran plot. There is one more most important method, molecular docking. So often I was describing the efficacy of a drug can be predicted using the binding affinity. By checking the binding affinity, the binding affinity can be determined through this molecular docking method. Okay, we have information, we discuss more information on molecular docking in subsequent sessions. Bonding and non-bonding interactions are most important to understand about that. Okay, so I think the other information on this, okay, we'll be seeing in the next sessions. Now, let me go to the websites to at least show you how to work on these tools. So can we go initially with the Swiss ADME? So if you want to work with the Swiss ADME, I told you this is the URL, swissadme.ch. Go to this kind of a tool. Okay, so what it's asking, it is simply asking you to enter a list of smiles. Where either you have to paste the smiles format, or even you can draw this chemical compound. We have a Marvin kind of a JavaScript viewer from Chem Axon. Simply draw a structure. Okay, and after drawing the structure, okay, click run. Or if you want to initially try with an example, fill with an example and you can run. Okay, but if you want to check specifically, want to check from the data generated from the database, you take a data from the database. Like how I mentioned you, in NCBI, we have a database called PubChem. Okay, this is a repository of chemical compounds, drugs, and inhibitors. You take any drug, whatever you want to check it. For example, if I want to check the aspirin, what is aspirin actually? Acetyl salicylic acid. Okay, if you want to get description on this, click inside. Now just type the aspirin, isn't it? Either type aspirin or whatever models you have, examples you have, click it and try with it initially. Now I get inside the aspirin. You are getting some detailed information on this, molecular formula, synonyms. So more information on that, description, more description on what that kind of diseases it cures and what kind of inhibitors you have. Here you have cyclooxygenase inhibitor, aspirin is a cyclooxygenase inhibitor. 2D structure information on that, and 3D structure information on that. So we can have, so only even if you want to relieve from body pain, okay, even we are also taking paracetamol, isn't it? So earlier we are taking the aspirin, but aspirin has some kind of a side effects. So even the patients, if have suffering from the body pain, defaultly they take paracetamol, like how they take for fever. Okay. Now, if you see this kind of a descriptors, okay, we have so many information here. If you want to get a 1D information, the canonical smiles we got like from here. Okay, we also have other kind of isomeric smiles that we should not take that. We have to take only the canonical format. So just type the canonical smiles format inside. Okay, even the 2D, 3D structures can be downloaded from there. Here it's not asking any structures, just the smiles format. So just typed it. Obviously, this is activated now. Run. Earlier it was not activated. Now click run. So it takes just seconds or to generate these kind of results. So what kind of results? A kind of a drug likeness information. We can get information on lipid schedule, drug likeness filters, goose filter is there, Weber filter is there. To know more about goose filter, just keep your cursor here. You see here you can have 4 different properties, but the values are something variant with respect to lipid scale. And go to the Weber, you find nearly 2 parameters are there. So there are physical and chemical properties described in each and every drug likeness filter. So based on that, they are telling whether it is having a violation or not, and if it is showing a drug likeness, they simply describe it as yes. Okay. So not only we can find the drug likeness from the various filters, but also we can find the information, physical and chemical properties of the drug. So for the partition coefficient, we have plenty of partition coefficient information available. And this is something related to the metabolism. Cytochrome P, whether it's acting like an inhibitor or not, it's showing the information on that. And I told you distribution, isn't it? If this is something related to absorption, the absorption you can able to find from here, gastrointestinal absorption. So generally, the drug, whatever you take, it should be a oral route. Okay, the patients prefer to be the form of a oral route. If the absorption is more, then obviously it's fine, isn't it? Especially for treating fever and cough and cold kind of diseases. So here it's high. So absorption is determined from here. Distribution, of course, can be rated through the blood-brain barrier thing. Okay, if it is yes, then obviously, if it is permanent, then it's also a lipophilic kind. Okay, it is also targeting the brain, but generally we won't prefer a lipophilic drug. We just prefer the hydrophilic drug. But this drug is also targeting a brain. So if you take much drug, obviously you have to face brain disorders. Okay, so distribution, metabolism, excretion you can find from the skin information. Okay, these are some information we got from the Swiss ADME. Like Swiss ADME, we also have something called ProAdmet. Okay, so that is pre-admit, not pre-admit. However, I found the properties here also. You can find the same properties, but even the toxicity is an additional parameter we can determine, and even the other parameters like these parameters also we can able to find from the IDMA prediction. Simply type the canonical smiles format and get the results similar to the Swiss ADMA, or simply draw the structure. Cover it as if for predicting toxicity, go to the toxicity. Just use the structure of the form or the smiles format, and you can submit it. Okay, draw the structures like this. If you know the structure, okay, what happened? The net is slow. Okay, so draw the structures and get an information on that. Okay, we have seen the Swiss ADME pre-admit, and what else we discussed? So we have discussed something about pharmacophore mapping. So go for the Zinc Farmer kind of a database. Okay. Yeah, so these are some of the online tools. And virtual screening, actually the docking I'll be teaching you during the docking session. Virtual screening also I'll be teaching you separately. So I am going to teach you virtual screening using Autodoc PYRX. Okay, and the virtual screening I told you, you're not going to check efficacy of a single drug with a receptor, but you're going to check the efficacy through checking the binding affinity of docking multiple ligands with a receptor. Okay, in the virtual screening, we also have a method called lead hopping, where if you have a standard drug, okay, we cut the functional groups and we can detach it, and we can also add a significant kind of a functional groups, and we can check whether this drug is binding efficiently with the receptor or not. If this drug is binding efficiently with the kind of a receptor, then I can also synthesize this kind of a drug, and I can try with preclinical and clinical studies. And there is also one more method called increasing high throughput screening hit rates. If you have a random combinatorial library is generated from medicinal chemistry laboratory, Then instead of synthesizing the compounds and checking with the binding affinity of a particular receptor, you better try with all this through the high throughput screening. Okay, there is a kind of high throughput docking through softwares. Checking the binding affinity is most important. Okay, we should whatever drug it's going to bind, it should bind only in the binding sites. If it's an enzyme, it should bind only in the active sites. So this kind of a binding sites information can be predicted through various algorithms. This is one such algorithm, flood fill algorithm in Discovery Studio Ligand Fit. Wherever the water is going to accumulate, we call that as a lipophilic binding site. And if water is not accumulating, but still we have a kind of a pouch like a binding site, we call that as a hydrophobic binding site. Hydrophilic binding site and hydrophobic binding site. The hydrophobic binding site is what we call it as a lipophilic. Okay, there is a plenty of information to be described in the methods. Okay, even in the latest drug discovery hackathon, the students have utilized molecular docking using Autodoc tool. Okay, and they have used virtual screening using the Autodoc Pyrex to crack this. We didn't use any commercial software, only through the freewares. Okay, there we have generated nearly 3.6 lakhs in winning this kind of a competition. We didn't even win up to the final stage. We just crossed a phase 1 alone. For that itself, we got this much amount of during the internship. Okay, there is a plenty to explain about the other methods. The other methods will be subsequently saying in the upcoming sessions. So participants, I am completed with a few methods of drug design. The few methods will be seeing in these upcoming sessions. Any doubts you have up to present? If you have any doubts, you can chat. Sir, full form of ADMET: absorption, distribution, metabolism, excretion, toxicity. QACR: quantitative, structure, activity, relationship. No, sir, very clear. Okay, thank you. Any other doubts? Box 2, very good. I found more interaction from almost all the participants. Thank you very much. And tomorrow, okay, join sharply at 7 o'clock or join 5 minutes earlier. I try to take sharply at 7 o'clock and complete at 8 o'clock. Just because today we have taken much time, I have taken a few time to compensate you. Thank you very much, student participants. Have a great day. Bye.